Okay, for question number 1, it gives us a normal chromosome that listed out in the inversion chromosome with its letters, and it says that, well, it asks us the question, what is the allele and centromere composition of the 4 products of meiosis if there is a crossover between S and T? So this is actually the original right here. So between S and T is where this crossover is going to be happening. So essentially what you're doing is cutting it right there and then recombining it to make these pairs. So over on this side we have the uppercase up until S, which is continued in this other chromosome with the T all uppercase, and then the opposite of that is right here with all these lower cases. Note that in the originals there's one centromere between, or for each one, but after the crossover, this top strand, because of where it was divided, has both centromeres, and this bottom strand has no centromeres. And that is question number 1.
Hey guys, this is Christian. I'm going to be doing problems 2 and 3 today. Starting with problem 2, part A. I wrote it all out, so I'll just read it to you with the blanks underlined. It says non-disjunction in meiosis 2 produces 50% aneuploid gametes, whereas non-disjunction in mitosis can produce a mosaic phenotype. Meiosis 2, it's having that non-disjunction in the second part. So half of them will be normal, half of them will be aneuploid, 25% being 2n minus 1, 25% being 2n plus 1. And mitosis talks about in the notes how it produces a mosaic phenotype. B, meiosis in an individual who has suffered a paracentric inversion produces a dicentric chromosome and an acentric chromosome. C, Turner syndrome is an example of a disease caused by monosomy, while familial Down syndrome is an example of a disease caused by a Robertsonian translocation. Turner syndrome is just the X. You only have the one sex chromosome. It's just X. There's no second X chromosome or a Y chromosome. So there's that non-disjunction causing that monosomy. And familial Down syndrome is different from trisomy 21 in that there's a translocation, that Robertsonian translocation, it says right here, between chromosome 14 and chromosome 21. D says human liver cells can be 4N, 8N, or 16N. Talks more about it in the notes. It's just talking about how there's benefits to certain organisms or certain cells in the human body as well, being polyploidy. E, terminal deletion in chromosome 5 causes Creutzfeldt syndrome, while an inversion on the X chromosome causes type A hemophilia. Pretty self-explanatory. It talks about in the notes how there's that deletion on the P arm of chromosome 5 that causes that syndrome. F says a duplication is a change in a chromosome structure that can be potentially beneficial to an organism or an entire species. Just more genes, more gene duplication of that gene helps produce more proteins to get whatever it needs to get done. G, when a translocation cross is formed, the alternate segregation pattern can produce viable gametes and offsprings. The other two types do not produce viable gametes and offsprings. Moving on to question 3. 3A is just asking total chromosomes in these syndromes, these diseases. Turner syndrome is 45. I talked about that. You just have one X chromosome. Jacob syndrome is 47. Non-disjunction causes you to have 2 X chromosomes plus a Y chromosome. Creutzfeldt, like I was talking about, you have all your chromosomes. There's just a deletion on part of chromosome 5. Familial Down syndrome, same thing I talked about. You still have your chromosomes 21 and 14. There's just that translocation. Type A hemophilia, you still have 46. There's just an inversion on X. So you still have X. It just has an inversion on it. B, we're talking about terms not associated with the Charcot-Marie-Tooth disease. So I circled them. I crossed this out, but then I realized I should have put it back. So terms that are not associated with it would be triploid, aneuploid, reciprocal translocation, trisomy, and non-disjunction. This is because the disease is just caused by duplication. So you're still diploid, you just have a duplication of one of the genes. C, terms associated with Klinefelter syndrome. That's the XXY, you have two X chromosomes, is associated with aneuploid, you have an extra chromosome. Trisomy, that's related to having 3 chromosomes when you should only have 2. And non-disjunction. Non-disjunction is what causes this. Terms associated with type A hemophilia, none of these are going to be associated with none of these terms because the disease is caused by an inversion on the X chromosome. None of these are related to a non-disjunction, monosomy, none of that. And that's all I've got for today.
Question 4. Not quite so easy, but it is if you look at the diagram. If you draw that out correctly. So, looks like a lot. It kind of is a lot. I ask for forgiveness for my poor art skills. Essentially what I have right here is in the red crayon that I used. The red is designating chromosome, the normal chromosome 1, and then just my regular pen color is number 2. So that's going to remain true throughout the rest of my diagram as well. And then the dots, or the little star blobs, are the centromeres. What it's asking, let me get to the question real quick. What this one is asking is for us to find, well, part A is just diagram the synapses of the chromosome in prophase 1. So that's what we have here, is this big diagram with the anaphase and everything happening down here. And then B, C, and D are asking us, we can use the diagram to help us with this. It's asking us to identify the allele composition of the gametes produced by, I guess, in the different segregations. So this side right here is the alternate segregation. I'll zoom in a little bit right there so you can see it better. Again, I know my art's bad, but the red is chromosome 1 and the black is chromosome 2. This is the alternate segregation. This is the adjacent 1 segregation, and this is the adjacent 2 segregation. So these are the different gametes that can be produced by the 3. And the letters that correspond to those can be found down here. So it's going to produce 2 of each, and these are the chromosomes found in B, C, and D, where it's asking for the alternate segregation, the adjacent 1, and the adjacent 2 segregation, respectively.
So I am doing number 5 from the group problems. Number 5 says that a cat has 38 total chromosomes. And for A, it's asking how many chromosomes a cat would have if it was triploid. So that's 3N. So if there's 38 total chromosomes, that means there's 19 pairs. So if you just do 19 times 3, that equals to 57. So A, triploid, is 57. For B, it's asking a monosomic for chromosomes 12, 38. So monosomic is just 2n minus 1. So if there's 38 chromosomes, I did 38 minus 1 and 37. So B is 37. For C, it is asking terminal deletion on chromosome 7. So terminal deletion is just a part of the chromosome being deleted, and so it doesn't technically remove any total number of chromosomes. So there's still going to be 38 chromosomes total for C. For D, it says the Robertsonian translocation between 17 and 18. There will be 37 chromosomes because one chromosome is going to fuse with another, and that is why one of those will be deleted. And then for E, it's asking for a reciprocal translocation between chromosome 6 and 9. So there's no gain or loss of genetic information. It just creates a break in a chromosome, and so that is, there's still 38 total chromosomes in E.